Hi, my name is Jim. I'm a student over at NIDES. This is my ANP12 protein synthesis presentation. Hope you enjoy. I present to you Protein Pathway, the new family fun adventure where you race through the molecular machinery of life. Build an mRNA strand, splice out introns, assemble amino acids, and fold your very own protein to escape into the extracellular matrix before your opponent does. Who will be the first to transcribe, edit, translate, fold, and turn genes to glory? It's time to play Protein Pathway. Game setup: each player chooses their game piece: polar bear, brown bear, Kodiak bear, black bear, or grizzly bear. Place all pieces on the start bubble, which is the nucleolus. Roll the dice, and the highest roller gets their first turn. Turn sequence. On your turn, roll your die and move forward the matching number of bubbles along the protein pathway. The player to your left draws a question card and reads it aloud. Correct answer: you successfully complete the cellular step and stay on your new bubble. Incorrect answer: oops, a molecular mishap. Move back one bubble and try again next turn. Mutation Mayhem, the red bubbles. Landing on a red bubble triggers a mutation card, a random twist in the genetic code. Some mutations speed you up, others slow you down, or even send you scrambling back to a checkpoint. Checkpoints of protein production. There are 4 key checkpoints on your journey. Start: the nucleolus, transcription checkpoints, DNA to mRNA, translation checkpoint, mRNA to amino acid chain, and protein folding checkpoint, final folding into a functional protein. If a mutation card sends you back to a checkpoint, you return to the last checkpoint you successfully passed. Think of these as your save points in the cellular process. Victory condition. The first player to bud out or exit into the extracellular matrix completes the synthesis journey first and wins the game. Now, it's time to play Protein Pathway. All amazing things must start small. With that being said, close your eyes and imagine a 15 million times smaller version of yourself. We are now small enough to explore the amazing world of the nucleus. Just like protein, we must start our journey at the nucleolus. The nucleolus is an area within the nucleus where ribosomal RNA or rRNA is synthesized and ribosome subunits are formed. As we leave the subdivision of the nucleolus, we meet up with our good friend, deoxyribonucleic acid, but their friends call them DNA. DNA is a self-replicating double helix material that carries all the genetic blueprints that are present in all living organisms. Consider DNA as the holder of the instructions of this very game. Without them, we would not know how to play. As the holder of those much-needed instructions, there is important information that DNA's neighbor ribosome needs to help them make a protein. But DNA is unable to leave the nucleus, so they need help getting that message delivered. This is where messenger RNA or mRNA becomes the postal service of the cell. Unfortunately, DNA's instructions are kept safe in DNA's vault and are in a language that ribosomes do not understand. This information must be first extracted and transcribed before a ribosome can use it. This sounds like a job for super helicase, an enzyme that knows the passkey to unlock the vault, and their sidekick, RNA polymerase. Psst, by the way, the passkey is UNZIPPING. Once in, the helicase begins at the initiation site. Using their superpowers, they break the hydrogen bonds between nitrogen bases. This elongates the vault walls, giving RNA polymerase a chance to transcribe the necessary information by creating an mRNA copy. When the information is copied, RNA polymerase triggers the termination site tripwire, instantly evacuating the newly formed synthesized RNA out of the vault as pre-mRNA. Now, out of the DNA vault, pre-mRNA must convert into mature mRNA. They start by putting on a 5' methylated cap and a 3' poly A tail. It is now ready to remove any non-coding sequences called introns through a process called splicing. Now that we have a mature mRNA, they are ready to deliver the instructions to the ribosome. Congratulations, we have reached the transcription checkpoint. Now that DNA's message has been transcribed, it's finally time for mRNA to deliver the important message to the ribosome. mRNA exits the nucleus through a nuclear pore, taking the rough endoplasmic exit straight to the ribosome. The ribosome consists of a small subunit and a large subunit, with the receiving bay located in between both subunits. During the receiving process, both subunits bind at the region of the mRNA with the methyl cap and start codon, which is made up of a specific sequence of nucleotides. Transfer RNA, or tRNA, arrives to accept the delivery. tRNA holds the anticodons matching the start codons of the mRNA. This whole delivery process is called the initiation complex, the beginning of a polypeptide chain. tRNA continues bringing amino acids to the newly formed polypeptide according to the complementary base pairing on the mRNA and the tRNA. Once the transfer is complete, the tRNA leaves the ribosome for a break. The ribosome then moves down the mRNA one codon length, and the new tRNA comes on shift with its corresponding amino acid. This process repeats, and the polypeptide continues to grow. At the end of the mRNA, there is a stop codon, which will end elongation. Consider this the end of the day school bell. Unlike the start codon initiating tRNA, the stop codon calls for a type of protein called the release factor, which will cause the entire complex of mRNA, tRNA, ribosome, polypeptide to go home, releasing the primary structure into the lumen. Way to go! You've reached a transcription checkpoint. Some proteins are transferred and float in the ER, while others are destined to anchor to the membrane. Our protein is to be secreted out of the cell. Before this can happen, though, the primary structure must fold into a full-fledged 3D structure, packed into a transport vesicle, then head straight to the Golgi complex. The primary structure has a particular order of amino acids joined by peptide bonds that dictate just how the protein is going to fold. It's like cars on a train. The order of train cars determines the train's journey and function. What do trains require to get from point A to point B? Tracks. As the train moves, the tracks twist into loops and ramps. These curves help guide the train along the path and give the first bit of shape. This is like the secondary structure. It forms localized patterns like alpha helixes, spirals, and beta-pleated sheets, folded tracks, due to hydrogen bonding between backbone atoms. Next is the tertiary structure. It's the full 3D folding of the protein, stabilized by interactions like hydrogen bonds, ionic bonds, hydrophobic packing. Just like a train, it now arranges itself into a full complex shape, like a roller coaster track folding back on itself, making bridges and tunnels. This layout ensures the train can reach the right stations, active sites, and perform its function properly. Some proteins have multiple polypeptide chain subunits that can join together to form one functional protein. Think of several trains linking together, forming a super train network that works as one big system to transport passengers or cargo more efficiently. Example, hemoglobin has 4 train engines working together to carry oxygen. After folding, the protein is packed into a vesicle for transport to its destination, inside or outside the cell. Like a train, at the end of the journey, the train loads its cargo into a special boxcar, which then detaches and sends off along a separate rail line, delivering the protein to the right city or cell location. Next stop, the Golgi. Wow, you're almost there! Welcome to the protein folding checkpoint. With the quaternary structure packed in transport vesicles, it travels along microtubules to the Golgi complex. Picture the Golgi as a massive Amazon fulfillment center of a cell, but instead of delivering books and gadgets, it ships out proteins and lipids. Now, to avoid copyright infringement, let's call our fulfillment center Golgiazon. Now, Golgiazon has receiving docks on the cis face at the receiving side of the Golgi. This is the first stop for our protein vesicle. Worker enzymes check each protein for defects, folding issues, or missing parts. Misfolded proteins may be sent back to the RAR for repairs. This all takes place in the cysticerny, Golgiazon sorting and quality control room. Next, we head over to the medial cisterny. Our protein gets special modifications like adding sugar chains, glycosylation, or phosphate tags telling them where to go in the cell. This is like a personalization station, where items are gift wrapped, engraved, or bundled for specific destinations. Our protein package is ready for labeling and address printing. This takes place in the transcisterny. Just like Golgiazon, each protein gets a molecular tag, like an address label, ensuring it knows whether to go to the cell membrane, a lysosome, or to be exported out of the cell. Remember back at the receiving docks where the worker enzymes check each protein to see if it has defects or misfolds? Those proteins go for recycling if they need more work. They either head to earlier Golgi compartments or go back to the ER. This is retrograde transport. Just like Golgi-Azon's return center, they process returns or reroute a package when something goes wrong. Both processes are crucial for the Golgi and Golgi-Azon by maintaining proper function and package/protein sorting. After bouncing around the trans-Golgi network, the TGN, a major secretory pathway between the subcellular departments of the Golgi, basically it's the warehouse conveyor belt system. It's finally time for shipping and dispatch. Vesicles bud off the trans face of the Golgi, each carrying carefully packed proteins to their proper destination. Delivery trucks, vesicles, leave Golgi-Azon loaded with packages heading out across the city of the cell, or outside of the cell entirely, to the extracellular matrix. Congratulations! You've budded out and are now on your way to your determined destination. Where is that exactly? Well, you're gonna have to wait for the expansion pack.